Okay, so we're going to talk about ATP and anaerobic metabolism. So a little bit of review here as we do this. So here are our objectives, or at least some of them. There's going to be another slide that has a few more objectives, but we're going to focus on ATP and ATP-PCr. Okay, so first is how do we produce energy? Okay, and really what we're doing, the better question to be is how do we use ATP for energy? So if we look at this chemical reaction here, we take ATP, which is adenosine triphosphate, okay, and that's just shown here. So we've got an adenosine molecule, there's a ribose sugar, and attached to that are 3 phosphates, okay, so tri means 3, right? And what happens is we break that down with the help of an enzyme called ATP-ACE. Whenever you hear or see something that has the suffix ACE on it, that's an enzyme, it's breaking something down. So we're breaking ATP down. We're breaking off this last phosphate, basically breaking this bond here, and we're left with ADP, okay, and then this free inorganic phosphate, and then we also release energy. These chemical bonds are actually stored energy. Okay, so I want you to imagine there's those gag gifts where like someone opens a soda and out of it jumps like that springy, snaky thing. That's essentially what's happened. When that bond is in place, that springy, snaky thing's just kind of kept inside, but once we open it, we break the bond, energy is released. Okay, now the body stores very little ATP, and this is because if we stored all the ATP that we'd actually need to function, we would weigh a lot. Okay, it would require a lot more energy to take on and to support the weight that would come from it. And it might seem a little crazy to think of because you're like, ATP, that's like a microscopic molecule. Yeah, but if we stored enough to be able to function on a regular basis, we would weigh a lot more. So because we store very little, we need to be able to produce it in times where we have high energy demand. And so we're going to go through several different ways that we can produce energy, produce ATP. And the first one is the ATP-PCr system. Okay, now, like I just said, we store a minimal amount of ATP, but we also store something called phosphocreatine. Okay, now when we break phosphocreatine down, that creates something called creatine. It also releases a phosphate, and when we break phosphocreatine down, remember there's a chemical bond involved there, we get this energy, okay? Now what's really interesting is that when we can take this energy that's been released and we can take this phosphate and this ADP and we produce a molecule of ATP, okay? And so creatine kinase is the enzyme that breaks down phosphocreatine. Okay, we have lots of creatine kinase when we have lots of ADP or phosphate concentrations because we're trying to take advantage of the fact that, hey, we've got this ADP, we want to put it back into or create ATP from it so that when it comes time to create energy or use energy, we can break that ATP down. All right, and so we've got this, and the ATP-PCr system, this is what you use immediately. Okay, so when you stand up, when you go for a run, when you start sprinting, okay, the first few seconds of an exercise, that's when you're using your ATP-PCr system. But it only lasts for a little while because we deplete, we remove, we get rid of, we use, whatever word you want to use, our phosphocreatine stores, and it only takes about 15 seconds. Okay, so for most of us, that's a little, we can run 100 meters in a little over 15 seconds, right? If we're, going at the Olympics, that's about 10 seconds or less than 10 seconds if you're a guy, a little over 10 seconds if you're female. And so they're relying primarily on the ATP-PCr system. Okay, so just to make sure that we understand, so creatine kinase is the enzyme that allows us to form ATP with the help of phosphocreatine. Okay, remember kinase, ACE, that means that's something that's an enzyme that breaks something down, and so it's breaking down phosphocreatine. When we have lots of ADP or lots of phosphate, and the I just means inorganic on the P, then we have high activity for that creatine kinase. But if we've got a lot of ATP floating around, the creatine kinase doesn't work as well. It's not as active, and that's okay because creatine kinase ultimately forms ATP. Well, if we've got lots of ATP floating around, we don't need more. All right, and so this is an example of a negative feedback loop. High ATP concentration causes a decrease in the activity of creatine kinase. That way we don't have a lot of ATP that we're storing. Okay, and this will lead us to glycolysis. Now, the reason we have ATP-PCr and glycolysis together is because this is anaerobic metabolism. In other words, it does not require oxygen. Okay, so we're producing energy without the help, or producing ATP without the help of oxygen. All right, so when it comes to glycolysis, there are 10 steps, and each of these 10 steps has its own specific enzyme that will take a starting molecule and create an ending molecule. Okay? Now, the first half we call the energy investment phase, and the last half we call the energy generation phase. So if you notice that at the beginning, we actually spend 2 ATP. In the second half, we produce 4 ATP, which means we net 2 ATP. So we'll start with glucose. We'll be 2 ATP in the whole. We produce 4 more, and so now we're plus 2 ATP. Now, an interesting or an important thing to know is that this process can either start with glucose or it can start with glycogen. Okay? And if we start with glucose, it costs us one of these ATP. With glycogen, we can just get rid of a phosphate. Excuse me, we can add a phosphate, and we can go to glucose 6-phosphate. Okay, so we can start with either glucose or glycogen. Glycogen allows us to produce one more ATP than it does if we start with glucose. So we go to phase 1, or the energy investment phase. Now, there are five phases to this, all right? And I'm not going to get caught up in, well, glucose turns into glucose 6-phosphate, which turns into fructose 6-phosphate, which ultimately turns into fructose 1,6-bisphosphate. We're not going to get into that, okay? But I want you to know that we spend 2 ATP during these first 5 steps. And basically what's happening is that we're just changing the shape and or rearranging things that are part of this molecule. Okay, so for example, we take glucose, and during this first step, we actually add a phosphate, and we get that phosphate by breaking down ATP. Okay, and then we go through a couple more steps, and we add another phosphate by breaking down an ATP. Okay, and so that's why it's costing us ATP, is that we need a phosphate, so we break down ATP to produce a phosphate. Now, ultimately what happens is glucose is formed into, or we create 2 molecules of fructose 1,6-biphosphate. Okay, and then, excuse me, we form 2 molecules of glyceraldehyde 3-phosphate. All right, again, you don't need to know each of these steps, but as you look through here, we produce some NADH that we can use later, and we'll talk about that with the electron transport. And then here, we form an ATP here, okay, and an ATP here, and then we form 2 more ATP at the end. So ultimately what happens is 1 molecule of glucose forms 2 molecules of pyruvate, okay? But that's only true if oxygen is available, okay? And if oxygen isn't available, we form something called lactate, which we'll talk about later on in the week. So this is occurring in the cytoplasm, or the cytosol of the cell. Okay, so that's just kind of the egg white part of the cell. And we're taking glucose, forming 2 pyruvates, and ultimately creating 2 more ATP. Okay. We need oxygen. If we don't have oxygen, we form lactate. And like I showed you a minute ago, we form NADH as we go through this. Okay. Now, remember, ATP-PCr lasts for about the first 15 seconds. Now, glycolysis is also occurring during these first 15 seconds of exercise. We're just not relying on it, okay? This is a 10-step pathway. 10 steps takes a lot longer than 1 or 2 steps, okay? And so glycolysis, once it gets going, it's going. And this is the primary energy system that we use during events lasting 1 or 2 minutes, okay? So if you like track like I do, or you like swimming, so if you like swimming, like in the Olympics, this is something that like anything that's a 100 or a 200. For the most part, those events last between 1 and 2 minutes. If we're talking about running, this is somebody doing something between the 400 and the 800. Okay, and as you go through lab, one of the things that we've done in lab is we actually are going to have you do a lab where you do events that focus on one of the three primary energy systems, whether that's ATP-PCr, whether that's glycolysis, or whether that's aerobic metabolism. Now, the last thing that I want to mention is something called phosphofructokinase. This is the enzyme that's responsible for the third step of glycolysis, and this is what we call the rate-limiting step, okay? And the reason why is it's just like it says here. If we've got lots of ATP, phosphofructokinase isn't very active. We don't need to produce more ATP if we've got a lot just floating around. But if we have very little ATP and or high levels of ADP, then phosphofructokinase activity is high, so we can produce more ATP. Again, this is an example of a negative feedback loop. And with that, we are done talking about anaerobic metabolism and the ATP-PCr system.